Thanks for inviting. I'm somewhat an outsider to this field. I work mostly in modern lake sediments, but I have got involved in the early Earth research through working with Sean Crow on Lake Matano, an Archean analog. And I had an enthusiastic PhD student who wanted to keep modeling this stuff. So I'll talk about how we interpret the sulfur isotope records in Archean sediments, and also what we may be missing currently from those models. So as was just presented, there is pretty good evidence for coastal oxygenation just before the great oxidation event, before the atmosphere got oxygenated. So we're interested in how this may have affected the sulfate concentrations, possibly methane fluxes as well, and how we can separate the signals that propagate from the atmosphere through the oceanic water column and get preserved in the sediments. So when we look at this sulfur isotopes, the typical trends that we see are that mass-independent sulfur isotope fractionations disappear at GOE, and the predominant paradigm for mass-independent fractionations is that ultraviolet radiation separates positive delta, negative delta into the elemental sulfur pool and sulfate pool. And these are incompletely mixed, so the positive excursions get preserved. The elemental sulfur can disproportionate, also generate some sulfate. But basically, we're looking at the sulfides preserved as pyrite in marine sediments. There is a relationship between mass-independent fractionation delta theta 3 and delta 34. So for a decrease in delta 33, that is probably a decreased role of elemental sulfur. We also see lighter isotopes in sulfur, which probably reflect microbial sulfate reduction. When we try to simulate preservation of these signals, we use diagenetic models, which importantly are analogous to the models that we use in modern sediments. That is, we consider these sediments as analogous to modern sediments where sulfate diffuses from the water column into the sediments. The sulfate reduction happens, and that's how we get our pyrite. We also use fractionation, microbial fractionations that we'll learn in modern systems with modern microbial capabilities. For the lack of better information. So this is a picture from Lake Matano, which has high iron concentrations. This is in the water column. You may as well consider stratified sediments. Sulfate diffuses in, produces sulfide. As sulfate diffuses in and gets reduced, we have Rayleigh fractionation, so sulfate gets isotopically heavier, sulfide must be getting isotopically heavy as well. The reason why we don't get exactly zero fractionation is that the system is open. So because lighter isotopes are preferentially used up in the sediment or in this deep water column, the gradients for the lighter isotopes are steeper, so there are more lighter isotopes diffusing in. We get essentially lighter sulfide. If we assume that at low sulfate concentrations, fractionations decrease because essentially all of the sulfate is being used up, we get low isotopic differences, delta 34S, between sulfate and sulfide. And we could match this with Archean pyrites for fairly low sulfate, very low actually, sulfate concentrations below 5 micromolar in this work. We can develop the model further. In a more recent paper, we included the oxidative cycling of sulfur, where sulfide can get reoxidized, so in coastal environments. Also put in disproportionation of elemental sulfur that wasn't really considered there. And what we found out is that oxygen, primarily limits sulfate reduction because organic matter mineralization happens to some degree through aerobically. This increases delta 34, through oxidative recycling, but paradoxically, it also increases preservation of mass-independent fractionation because you get less sulfate reduction, so more signal gets carried through from elemental sulfur. If we put this in the context of sulfur isotope records, what this seems to explain is the increase in delta theta 3 leading up to the GOE, coincident with the increase in delta theta 4. And if we try to put constraints on the sulfate concentrations that's consistent with this, it looks like the sulfate concentration increased from about 50 micromolar. We relaxed the previous constraints on sulfate concentration to about 50 micromolar, up to about 200 micromolar. And oxygen concentrations that could reach, 25 or so micromolar in the shallow environments. To reproduce these trends, this also seems to suggest perhaps increasing organic productivity, increased deposition of organic carbon. If we carry simulations further in time into the Proterozoic, we could look at the sulfur budget in the Proterozoic ocean. So in a fairly simple model that considers coastal ocean, surface deep ocean, and deep ocean. For realistic fluxes, input and output fluxes, what we seem to have is that sulfate concentrations were in the hundreds of micromolars. They were not as high as previously believed. The important result from the sensitivity analysis is that the sulfate concentrations in the ocean seem to be controlled by the presence of this anoxic, huge anoxic compartment. If you oxygenate the water column, this increases the sulfate concentrations tremendously by removing this deep sink, and this is how you can get millimolar or higher levels of sulfate. But until the ocean, deep ocean becomes oxygenated, sulfate concentrations may have remained fairly low. So this figure basically addresses this. There are some exceptions to this result. So Loma Gundi seems to have high sulfate concentrations, which may suggest that the water column have been oxygenated. These have been suggested in the literature. Diagenetic models show that basically these sulfate concentrations are consistent with sulfur isotope records if we consider tens of micromolar of oxygen in the surface ocean. At these low sulfate concentrations, methane fluxes are sufficient to maintain concentrations above 25 ppmv, which have been suggested as sufficient to maintain the Earth's glaciation free. Okay, so I have, in the remaining few minutes, I'd like to talk about the factors that we may not have put into these models. Like I said, previously we considered sulfate diffusing in from the water column. That was the primary source of sulfur to the sediments. Well, if we look into the modern lake sediments, low sulfate lakes, sulfate or salt and sulfide seem to be generated within the sediment from organic matter. Well, these are the plots from Lake Superior, which is oxic, and Lake Malawi, which is borderline ferruginous, sulfidic. Sulfate concentrations in the top layers of sediment are actually above the layers, the levels of sulfate in the overlying water. So sulfate actually fluxes out of the sediment. While in Lake Matano, CRS fraction, which is basically pyrite plus elemental sulfur, increases, but the total amount of sulfur actually decreases. So we have a conversion from non-CRS fraction to CRS fraction, and this decrease is consistent with the organic sulfur being mineralized, and so the ratio of total sulfur to total organic carbon remains remarkably constant within the sediment, which again suggests a role for organic sulfur. Now this has a profound effect. Instead of considering Rayleigh distillation in the sediment column or water column, we have sulfate and sulfide being generated within the sediment, possibly with the isotopic effect of organic sulfur hydrolysis. And so this changes the paradigm of how we generate the isotopic signatures in sediments. If we construct a model that simulates how much pyrite you can generate, turns out you can generate at low sulfate concentrations, you can generate all pyrite in the sediment from organic sulfur. And so if you consider organic sulfur in organic debris, prokaryotic cells actually lack +6 like sulfates, but they do have sulfonates, which is +4. And of course, they have -2 oxidation state. So you generate both sulfide and sulfite from mineralization. And sulfite reduction is actually energetically more favorable than sulfate reduction. So if you form pyrite out of this source, the resultant signatures are also consistent with the rock record. They're also consistent with some other lines of evidence that show, well, sulfate reduction has evolved later than sulfite reduction, in fact, as late as 2.7 billion years ago. And this also seems to suggest that you don't need as much sulfate in the water column. So if you look at the sulfur to carbon ratio in living cells, it's pretty similar to phosphorus to carbon ratio, about 1%. So at these levels, sulfate could have been a limited nutrient for life. Depending on what you believe about Archean productivity, mineralization of organic sulfur could generate enough sulfur to account for global pyrite fluxes. And it's comparable to the flux of sulfur from the hydrothermal system, or actually exceeds that flux. So organic sulfur probably needs to be considered. If you try to forecast the effects on the isotopic signatures, this is delta 33, delta 34, depending on what you believe about the assimilation of organic sulfur into the organisms, it probably falls on the continuum between elemental sulfur and sulfite. The microbial reactions would shift the resultant pyrite to the left here. Interestingly, around this area, we find sulfides in microbial mats, which again, organic-rich system with abundant organic sulfur. I have just a few more minutes. I want to briefly comment on other factors, other uncertainties in our interpretations. We don't really know what happens at low concentrations, and low concentrations and low rates are not the same. This is a figure from a recent review that showed that these are the bars are the cell-specific sulfate reduction rates in modern marine sediments. These are the data points found in modern experiments. We lack data on low sulfate reduction rates, specific sulfate reduction rates. Low reduction rates, however, cell-specific reduction rates, are not necessarily associated with low concentrations. Below about 500 micromolar of sulfate, sulfate reduction rate starts being limited by sulfate, but organic matter is still important. So when you look at the sulfate reduction rates in lakes, where concentrations of sulfate are just tenths of micromolar, they're not that different from marine sediments, where concentrations of sulfate are 1,000 times greater, because they're controlled mostly by organic matter. So we probably don't need to look that deep into marine sediments to those low cell-specific rates. And finally, we have to be conscious of the sampling bias. The sulfate reduction in modern sediments happens predominantly in coastal sediments, and coastal sediments are extremely heterogeneous environments. If we try to say something about modern environments by looking at the modern ocean at just a few locations and try to extrapolate to the entire planet, we probably would have been laughed at, but that's essentially what we do for the early Earth for the lack of better alternative. I think we have to be conscious of that. And I'll end here.
sulfur going from the sulfate reduction pathway or disproportionation. How is the sulfur complex to organic matter once it gets out of the cell? Does that matter in terms of the isotopic signature, fractionation, or the way it's processed? In other words, it can be bound to different types of organic functional groups.
Right. I think it's very important, especially which step during mineralization is rate limiting. We don't have that much information on how sulfur gets fractionated. There is some information primarily from lab studies, but I think that's what we need to research, how these organic compounds get processed inside and outside the cell.